Bold is breaking barriers, pushing beyond the known. Innovations born from courage, making the impossible shown. Bold is opening new horizons and seizing opportunities every day. New technologies and therapies, improving lives as we lead the way. Bold is delivering on the promise of treatments both safe and sound, where the patients come first, where compassion and expertise abound. Let us boldly advance innovation every day, for innovation that matters to patients must be bold to lead the way.
Please welcome WuXi, Program Chair and Vice President, WuXi AppTec, to kick off the online event.
Good morning, good afternoon, and good evening, everyone. Welcome to the second episode of WuXi AppTec digital program, Bold Innovation That Matters. Through this series, we'll bring together leading organizations at the forefront of breaking barriers, opening new horizons, leading next-generation therapeutics, and delivering on the promise to patients. We believe that bold action and bold thinking are essential to drive meaningful benefits to patients. Today, we'll hear from 30 leading voices on what bold means to them and how new technologies and approaches are redefining drugability and advancing new modalities. At WuXi AppTec, we're committed to fostering innovation as a dedicated enabler, a trusted partner, and an active contributor to advancing breakthroughs for patients. By working together, we can think boldly and act boldly in addressing global health challenges. As we start today's program, I would like to express our deep appreciation to the esteemed speakers for your insights and inspiration. And thank you all for joining us online today for these important discussions. Let's tune in now.
Thanks to remarkable scientific advancements in our understanding of diseases and biology, treatment options for patients have rapidly expanded over the past decade. How can we harness the power of breakthrough science and technologies to better address global unmet medical needs? In what ways can we leverage bold approaches to accelerate drug discovery and development? What does bold mean to our industry in the next 5 to 10 years? Join us to hear from our esteemed panelists as we delve into these topics.
Thank David, James, Matt, and Sabine to join this session. So as a background, this session is really, we are kind of talk about bold, right? So the dramatic advance of our understanding of disease biology has led to an increase in treatment options for patients at an outstanding rate. As a result, the past decade has seen a surge of new modalities encompassing genetic medicines, precision approaches, among others. So during our today's panel discussion, let's explore and discuss how bold approach may foster innovation and collaboration in our industry to prepare our industry forward in pursuit of better medicine for patients in need. So that's kind of overall arching theme for this panel. Maybe let's start with first question is just everybody share your view. So what are the top breakthroughs over the last decade, and have they improved patient healthcare and outcome significantly? And what are the biggest unmet needs that still exist today? So feel free to share your view with the audience.
So maybe I can start, being the one with the less background in science. Well, I was fascinated about how much the better understanding of the immune system broke certain barriers, starting with obviously oncology, checkpoint inhibitors, immune cell therapies, which transformed some patients who were totally hopeless out of any therapeutic option, giving them new hope. And that is now coming into autoimmune diseases, where we start to see a refractory resistant patient to first lines. So maybe one question would be, would we see in the future, while we are progressing in understanding how this fascinating immune system works against or for, will help us to understand more about the CNS, which I see as one of the barrier to crack. More and more, we see the evidence of an immune component in the neurodegenerative diseases. And I do feel that this is a field where we really need to progress. Will that come from this better knowledge of the immune system? That's what I may ask to the audience, and I hope we can overcome that next barrier.
Okay, great.
Yeah, I certainly would agree with you, Sabine, about immunology. First of all, Ed, thank you for the invitation to be here today, and it's wonderful to be among these terrific other panelists. Yeah, I would agree that over the past decade, the breakthroughs in immunology certainly have been one of the most exciting things. I think what has really been impressive for me over the past decade has been that every one of the types of medicines that we use for our patients, whether they're small molecules, proteins, or now cell gene therapy and gene editing, all of them have had pretty exciting breakthroughs over this period of time. We're going after undruggable targets with small molecules based on our ability to visualize novel pockets on these molecules, making it easier to find these important small molecules. I don't think small molecules and pills are going away. I think they still will be an important part of our armamentarium for patients. The advent of bispecific antibodies to bring, to your point, Sabine, T cells to tumors and other indications. And then, and Jim is the expert here, the revolution we've seen in cell and gene therapy has been just remarkable over the past decade. As a hematologist, if you would have asked me in my prior full-time practicing career that we could cure patients with sickle cell disease or thalassemia, or it just would have been unimaginable. So I think we've seen incredible breakthroughs in all of the classes of medicines, and we've created whole new classes of medicines. Certainly a lot of challenges we'll get into, but for me, it's the breaking open of the biology as well as all the modalities moving forward.
Well said, David. James, do you want to comment? You are a pioneer in the cell gene therapy field, and you certainly witnessed how our industry kind of evolved.
Sure, I'd be happy to. So this gives me a chance to reflect a bit on my career, which spanned more than 10 years. So I'm a little older than that. But I guess what, as I do reflect on one part of genetic medicines, which was gene therapy, which is something I started to study 40 years ago, was really how long it took. But as I also think about what we knew when we started this field, we knew very little about basic fundamental biological principles. We didn't even know about co-stimulation and signal 2 when we started in vivo gene therapy. So the one thing that is exciting to me as we stand up new platforms is not only the power of the platforms, but the incredible insight that we have in basic human biology. And so as I think about the last 10 years, I think about new platforms, you know, as we witnessed with RNAi, mRNA beyond vaccines in my world, which is rare diseases and genome editing, that I expect that these modalities that either were discovered or were beginning to be translated, that the pace with which we can move these forward because of our understanding of biology is going to be substantially faster. With that said, when you get into human experimentation, it naturally has a cadence that some are impatient with, and there's only so it's only so fast that we can move as we enter into clinical trials.
Thank you, Matt.
For me, I think the breakthroughs come in sort of different categories. The first is the breakthrough in immunotherapy for cancer, and the now earlier and earlier use of immune checkpoint therapy transforming the lives of many patients. I think the second thing is the area of medicinal chemistry and the efficiencies that it can now be achieved to engineer new drugs, even against old targets like estrogen receptor. So newer generations of highly engineered, very specific small molecules is here. And in the third revolution is in the area of cellular therapy for cancer, where we've broken a number of barriers to achieve the effects that we're seeing with CAR T-cell, now not just in heme malignancies, but in common solid malignancies. So I'd probably put those three things up there, along with the issue of drug delivery through antibody drug conjugates.
Okay, great. So every panelist talk about great achievement breakthrough already had in the last decade, but I also mentioned about the challenges. Right? So can you kind of elaborate on the challenges our industry is still facing today? And you can talk about technological challenges and non-technical hurdles to those challenges, including like delivery technology, right? So technology like mRNA or even gene therapy, those delivery today is still quite a challenge, right? And how do we, as an industry participant, is going to overcome those challenges over the next decades?
And maybe I can comment on delivery for genetic medicines. I think the first thing we need to do is to be smarter about the deployment of these genetic medicines for the applications. And in the past, there has been a misalignment as to what the delivery system can do and what we're asking the delivery system to do. So if we're more thoughtful about the early applications, I don't think delivery will be a problem. And it's really helpful to have clinical successes and commercial successes. With that said, once we are more thoughtful about how we proceed, there will be huge gaps in the applications that relate to delivery. And so there's a lot of conversation and work around non-viral delivery systems for large molecules, nucleic acid molecules. And LNP has surely done a great job in delivering mRNA for vaccines, but there still is a lot of work in that area, but a lot of investment in that area. So I hope that we will begin to see some progress. But even when there is a breakthrough at a basic technology standpoint, it's going to take quite a while before that ends up in the clinic and ends up in a commercial product. So I think we have to be patient. And now I'm speaking to the investors, if we could be somewhat patient and not just cut us off at the knees here, that there will be progress.
Just to build on Jim's comment, I mean, I would agree that we are seeing really important advances in delivery of the next gen, whether it's gene editing or other gene therapies. I still think one of the challenges we have, and I personally think it's an engineering challenge, I don't think it's going to take a scientific breakthrough, is how to deliver to certain cell types really specifically in certain tissues, which have been a challenge to date. We've handled the liver really well. We're getting very close to hematopoietic stem cell, but muscle, CNS are still a challenge to get to the cell type of interest. I'm pretty confident that we will get there, but I'm curious what everyone thinks, because I think one of the challenges we face with these exciting, potentially curative medicines in the cell and gene therapy, gene editing space is going to be access. In how do we get access to the broad patient populations? And right now, at least in the US, there's still such a diversity of access to even conventional therapies, let alone cutting edge therapies. And when we think of diseases like sickle cell and thalassemia around the world, as we have curative therapies that are coming close to approval now, how do we get them to the patients around the world that need them in an equitable fashion?
Well, maybe I could comment on that since we're sort of in the middle of some of those challenges. it turns out that the easiest targets are monogenic diseases, such as what you mentioned, sickle cell, and some of them are often quite small, and as everyone is aware of, there is a business challenge of how you price if it's a one-time delivery. So the way that I think about these one and done treatments or editing, gene therapy, is that we really have to think very differently about the way that we regulate and commercialize these products, and to drive down the costs in every conceivable way we can, and to leverage the power of the platform. So if we can drive down development costs if we can edit a gene in a hematopoietic stem cell for this disease, do we have to go through all of the same kind of work where we just swap out the gene?
And maybe if I can chime in here and build on what you just said from indeed more the commercial piece. And it's indeed not only about the cost of R&D for sure, but there is a question based on, and the bridge is easy to build based on the breakthrough that we saw before. Now it brings also challenges in terms of scalability, reliability, easy to manufacture, point of care, because if this doesn't improve, now it's not only about R&D. And my second comment would be, I mean, and it's clear in cell therapy, as I think David mentioned. And my second could be, as I like very much the bold approach, it's all about probably collaboration. I do understand and accept a challenge for investors. However, if we really want to give the large access, we have to come together. Investors like JATO will be key, but as you mentioned, regulators, payers, patient advocacy will be major to make that happen. So it's not only about finance, it is an ecosystem. And I like in the bold the collaborative approach around the patients and their family to make things happen. So beyond science, there are some infrastructure and some collaborative platform to build, probably integrating deeply patient advocacy groups.
Maybe I can follow up on that a little bit. I think one of the areas that could achieve some of these goals is in the area of the diagnostics industry and delivering diagnostics in general. I was just about Jim's comment about, inborn errors of metabolism and their rarity. Sort of cancer is the opposite, isn't it? It's a sort of, chaotic genomes, hundreds of genes are affected, evolving all the time in your body. Different stages of cancer will have different somatic gene profiles. A hard thing to wrap your head around. But I do think there are a few things that are helping here. I think one is the advent of computational pathology. So there's a beginnings of a revolution from pathologists looking down the microscope to this being done by a computer where you're compiling that data very rapidly. So basically what that might mean is that a test would be done, the image would be logged into an artificial intelligence platform, which would spit out a pathology diagnosis of high quality for every patient. The other thing that's happening is we're converting simple techniques like immunohistochemistry from at best semi-quantitative activity into something that is fully quantitative by using mass spec calibration. So we know exactly the range where that stain is quantitative and the range where it's saturated or is at the lower limit of detection, and using that as a way to improve a simple diagnostic technique. And then, of course, the incredible decline in the price of next generation sequencing, allowing Jim to find those patients rapidly, and for me to find the somatic mutations that I'm going to attack, either using immune system modulation or genetic approaches and synthetic lethality. So I think that has to be brought into the conversation, because I think that diagnosis and diagnostics in general are also undergoing a revolution.
If I could just build on Matthew's comment, because I think it's a great one, because I do think, if I think of the last decade, one area where maybe as an industry, we maybe fell down a little bit and could potentially make even more of an impact over the next decade is in the space that Matthew's talking about. Because still many of the therapies we're giving patients outside the genetic medicines, which are tailored based on the genetics, obviously, still most of the standard of care, certainly all the immuno-oncology, the immune medicines we're using for cancer patients, we still don't have a predictive diagnostic to know which are the right patients to treat. So we're treating 100 patients to benefit 20 or 25, and that's just not tenable. The other areas, we've spent very little money as an industry thinking really creatively about how to tackle prevention. Not just in terms of environmental exposures, but can we intervene really early based on whether it's clonal hematopoiesis or other ways to identify patients much earlier in their particularly in cancer, but in other diseases as well, find novel targets and intervene to prevent, rather than spend our whole careers treating patients who are at sort of end stage and then finally a decade later working up into the earlier stage patient. But start in the earlier stage patient and make a bigger impact.
Just let me expand on that in sort of the very earliest of diagnosis, which is an area that is, I think, going to substantially grow and change the way we approach genetic disease. That's newborn screening. So there currently is newborn screening. It's limited, maybe 35, 40 diseases. It's really supported when early intervention makes a difference. And if there isn't a treatment at all, then there isn't much motivation for newborn screening. But an example would be spinal muscular atrophy type 1. 5, 6 years ago, there were no treatments. Now we have 3, and potentially a genetic treatment, a one-time treatment. And if we wait for those patients to be symptomatic when we intervene, the outcome is very, very different.
So also want to build on Matthew and James and David, you have excellent comments about diagnose patient better and then treat them better. But given today's diagnosis for those, particularly for cancer and those diseases are quite sophisticated. So only a limited center can do right properly. So if we want to scale this technology and allow not only United States, but other countries can also be able to access. One of the technology, obviously today, everybody talk about is the AI technology, machine learning. So can we take those learning into the AI? So the AI technology can assist even less experienced physicians be able to diagnose properly and then treat those patients properly.
It's very interesting to me as we have this very sort of DNA-centric look at the world. But of course, a cancer phenotype is executed by proteins, their post-translational modifications, and by the metabolome of the cancer. And we don't measure any of those things routinely. But in terms of AI, what we're seeing now is efforts to integrate information from multiple omes. So DNA, RNA, protein, post-translational modifications, and the metabolism to get a holistic picture of that cancer cell and the way it executes its unfortunate biological effects. And we say, well, how can you turn that into any kind of diagnostic technique? Again, what we've seen over the last 10 years is also a revolution in mass spectrometry. At this point, we can get 7,000 or 8,000 proteins out of a paraffin-embedded block using laser capture microdissection of just a few millimeters squared of that cancer. And also using targeted mass spec as part of that paradigm, we can look at incredible accuracy. We're talking about 10 attomolar sensitivity for analytes of interest in panels. And the thing about the mass spectrometry revolution is cheap. It's much cheaper than genome sequencing because the machine itself is expensive, But there's no reagents in the end, except a few spiked in heavy peptides, which are cheap as chips because you need femtomolar quantities of them. So the average mass spec S is about $40, right? And I'm not talking about, you know, the prep, but even the prep is now artificial intelligence guided laser capture microdissection. So it doesn't need a pathologist to grab those tumor cells. It's all computational pathology driven. So I think we're looking at a number of different diagnostic technologies. That's just one example where we can get a more holistic view of the cancer cell, screen many targets very accurately at once, and most particularly right now, surface targets for T-cell engagers, antibody drug conjugates, CAR T cells, and beyond. So I just wanted to put that in there because I think just looking at DNA is not going to be the answer for cancer, pretty obviously. Yeah.
I mean, certainly machine learning is playing a role in how we think about the entire drug discovery to development to delivery paradigm. But I think it's important to remember, I personally think the discussion of ML and AI in drug discovery, drug development is still a bit overhyped right now. Look, we've been using machine learning in drug discovery for 40, 50 years. Many of the technologies of machine learning that, and I'm not a machine learning expert, but I work with a bunch, those technologies have been around for a long time. They're not all that new. And I think it's a bit naive to think that you can use one of the AI programs and suddenly you're going to make a medicine in 2 weeks instead of the 5 years it takes now. Will it impact every aspect of how we think about drug discovery, drug development, clinical trials, access, finding patients? For sure. But I'm not sure it's an overnight revolution. But I do think we will incorporate it. And I think particularly as Matt was saying, being able to crunch huge amounts of data, having these technologies definitely helps, whether it's clinical data, whether it's pathology data, or even how to find patients for rare disease therapies. We're going to see advents of that. And then your last comment, Ed, I personally would still like to go to a human as my physician rather than a computer. I'm not ready to swap that out yet.
Yes, great.
Yeah, that's really great comments because we are, as investors, overwhelmed by this very crowded field, everyone bringing their own perspective. It's not the holy grail for sure. It will impact, but for us, it's not clear yet on what is a clear value proposition and how all these companies are differentiated one from another. Maybe having a big and holistic company like yours, Ed, would help navigate. As of today, it's still something very appealing, but still something a little bit vague for us, and we have difficulties to figure out. However, indeed, in clinical development, every help to make the translation more predictive and the clinical development faster and less expensive, not in 2 weeks, obviously, but more predictive when you will go into real life for patients, that's helpful. But for us, indeed, it is a question mark. We investigate, we try to understand, but not clear value proposition yet.
So one of the biggest costs in our industry is actually clinical trial failure. And often cases is because the target, you thought it's the relevant target, it end up being not very relevant in the disease setting when you run into clinical trial. For example, in the whole immuno-oncology space, right, there's so many checkpoints. But in the end of the day, so far only PD-1 is a really big hit. And then for CTA, the second. And then you have a bunch of other things, right? So far, we have seen very limited success. So therefore, the understanding target and disease biology is very complex, and traditional biology research takes a long time to figure that out. So I just wonder whether the AI technology, given its ability to handle multi-omics data in grand scale, where it will be help us nail down relevant targets, right, in the clinical setting. Right.
I wanted to follow up on that because this is a particular passion of mine. We don't study the reasons for failure of the standard of care adequately, right? Every day, patients are prescribed cancer drugs and they have a success or a failure for their own particular situation. And we're not tracking those patients and trying to understand the molecular basis for the outcome. So one of the programs I've started is called PathCR Predict. So basically, as you know, very often we give cancer drugs before surgery, and then there's a happy patient whose disease is eradicated at surgery, and then there's a patient who's unhappy because they've got disease still persistent, and those patients to go on to do badly. So imagine a world where you could actually tell the patient before they receive the drug which category they're in, the happy patient or the sad patient. Because if they're a sad patient, they've basically got an orphan disease. The standard of care is not going to work for them. And so you can be very innovative for that patient. And that also has another knock-on consequence, is that if you have those orphan patients in whom the standard of care is not working, you can do very small clinical trials to show progress, maybe even just single-arm trials to show that suddenly you've got a transformative therapy that will induce a PathCR in that patient, and they don't have to wait for the failure of the standard of care to get access to that drug when you've beaten up their immune system with a whole bunch of inappropriate drugs that don't work and it's suppressed their immune system. So I think that there's just millions of patients literally out there getting the standard of care, and we should study them. And I think that really would accelerate access to innovative medicines. I think we're moving beyond the idea that, you know, we need to do a randomized phase 2 trial with 1,000 patients to see a 5% increase in outcome and get approval in this huge population. That's too risky, too expensive, and essentially almost morally inappropriate these days. Study the biology, predict these outcomes, and then do more efficient trials in the unmet need population.
But Ed, I would agree with that. And Ed, to build on your comment, I mean, I think, you know, there's nothing more important in my view in the drug discovery, drug development process than increasing the probability of technical success. That outweighs all the other factors, in my opinion, that one considers when they're working on a program. And, you know, there's no question that one of the ways to do that, and that's been very fruitful, are going back to the genetics. I mean, when we identify, and people have identified protective variants in the normal population based on large genetic screening projects, and then determined that they have an outcome such that if we find that target and can drug it, we'll get the same outcome in patients with a disease. That's happened in cardiovascular disease, PCSK9, et cetera. So, you know, I think there are more and more of those types of genetically driven targets. I do see a lot of companies, and Sabine sees them as well. On the investing side, we see a lot of companies who are pitching, we're using AI to discover a novel target, not based on genetics. And then they don't do a lot of the traditional validation work using genetics and other tools, but rather want to jump right into the clinic because quote unquote AI has said this is a good target. And I'm not sure that increases the probability of success.
So I want to switch topic about the next panel is going to talk about the druggable target space. So in our industry, you know, certainly in the last decade, we are because of the new modality, we open up somewhat target space that are previously undruggable, right? Like protein degradation, right? Oligos, and obviously cell gene therapies, and those previous undruggable targets become targetable today. That opens up a new field for therapies to be developed. In kind of your view, for those type of therapeutics, right? Which kind of therapeutics will be most beneficial to patients that today is really untreatable, right? And then again, what will be the limitation, the challenges to overcome, you know, to bring those therapies into the market? What do you see, and how do you see the field evolving over the next decade?
Maybe I can comment on the potential for nucleic acid therapeutics to be able to address targets that are difficult to drug in a more traditional way, and it has to do with the specificity of hybridization, whether it's an ASO or an RNAi. I think it holds some potential there, but the problem is is delivery. So I think that's going to substantially diminish the advantage that we have possibly in expanding the target druggable pathways until we crack the delivery problem. And there will be a small number of target cells or tissues where delivery isn't a problem. But until we solve a delivery challenge, the potential of nucleic acid therapeutics and expanding targets is going to be limited.
I think, Ed, that, well, first of all, I would argue that the class of drugs, the IMiDs, which are approved and have made a big impact, are degraders, right? They degrade through cereblon. And I think there's a reasonable chance over the next decade we'll start phasing out the term undruggable target. Because there are a lot of new modalities, whether it's our ability using computational tools to find pockets for small molecules on targets that have been very hard. I do think the degraders, which come in so many different flavors now, will continue to make advances. There are a bunch of new modalities that are coming forward to try and drug protein-protein interactions and other what have been classically called undruggable targets. I don't think they're all going to fall down one by, overnight, but it'll take time. Look, a couple of years ago, everyone thought it was impossible to drug KRAS with a small molecule. Now, the first generation drugs maybe are not perfect, but clearly we've cracked that open and that will continue to crack. So I'm pretty optimistic that this class and other classes like it will make a big impact on the quote unquote undruggable targets.
Yeah, so I grew up in the world where everything had to be a genetic hypothesis. But these days, the surface proteome of the cancer cell is a huge opportunity for drugging. But in a very specific way, you're actually latching onto that surface proteome, your drug via an antibody, which allows all sorts of things to happen. But if you look at AstraZeneca's pipeline right now in cancer, probably 80% of our pipeline is drugging the surface proteome. And so those aren't genetic hypotheses. Those are really just cancer cells have stuff on their cell surface that's differential to a normal cell that you can latch on and deliver stuff. And I'm just fascinated by this idea that, we can move beyond the, I mentioned this earlier, the sort of, hey, let's just attach an old-fashioned chemotherapy drug that was developed 40 years ago onto that antibody, to all this innovation in this drug space, and maybe even deliver PROTACs that way with an antibody drug conjugate. Maybe some of these clever oligonucleotide approaches, maybe some small molecules that got left on the shelf were hyperpotent but couldn't be delivered. So I think that's another area that we have to think about. Of course, those surface targets are also massively important for CAR T-cell engagers and other forms of immune engagement. So that's just in terms of what's the drug target. We can't miss the surface proteome either.
Clearly, we see so much progress made already, Matthew, in your space of a surface target. All the CAR T-cell therapy requires anchoring the surface biomarker, right, in order for the T-cell to kill the target. So that's certainly very exciting. I want to come back on the earlier conversation we said. Still a lot is how can we deliver the medicine precise to the target cell they're supposed to go to, right? Rather than go to the whole body. Particularly for CNS disease, you want the drug going to the brain and that brain cell, rather than stuck in the liver or in your bloodstream. And do you see any really cool technology emerging that literally have like specific cell targeting capability, so deliver the drug to where the cell is supposed to be?
We are definitely seeing that, whether it's using, as Matthew said, whether it's using antibodies that can target things like the transferrin receptor to bring in small molecules, enzymes, oligonucleotides into the brain. There are a variety of people now using ligands, antibodies to drag AAV or lipid nanoparticles into specific neuronal cells across the blood-brain barrier, or need to be injected into the spinal fluid into specific neuronal cells. There's a whole series of technologies that are developing. I can't say, in my opinion, I don't think they're fully there yet. They have some early proof of principle, but it's certainly going to be the next wave. To what Sabine had mentioned very early on, neurology is still, and neurodegenerative diseases, neuropsychiatric diseases are still way behind where everybody else has been. And that's one of the hurdles. And there's a lot of work, but I think we'll get there, but it's going to take some time.
And that could be one of my questions to the high-level scientist you are. That do you see bridges? Because we talk a lot about immuno-oncology, about oncology, and there are so fascinating things happening, more and more targeted, more and more narrow, specific, with all the challenges we talked about. But do you see that learning from there will impact the enormous challenges of Alzheimer's and other neurodegenerative diseases, or there is no way to learn from one disease to another? Because we are almost now targeting and customizing patient per patient, while there are huge populations which are still just to bit on what you said on that CNS part, which are still without option as of today.
Just one comment about that. I mean, we think a lot about targeted, quote unquote, delivery, but we break it down into two barriers. One is gaining access to the cell, and then once there's access to the cell, attaching to the cell or getting into the cell. And in the central nervous system, I think the biggest challenge is not a tropism or getting into the cell; it's getting to the cell. Same thing for other heart and skeletal muscle, at least for larger molecules. And there are physical barriers to overcome that. Transferrin receptor may transcytose across it. And there is a lot of effort and a lot of progress. And there are precedents, at least in model systems, where this could be done remarkably well, not applicable to humans, but if it's demonstrated in a model system, then it's conceptually feasible in larger organisms such as humans.
Thank you. Because of time limitation, we have one final question, and this is very open-ended. So each panelist, please tell us, what do you think the next breakthrough is on the horizon? In the next decade, what do you forecast as a major milestone that our audience should be watching for?
I'm happy to jump in. I've been spending, and hopefully Jim will comment on this as well. I've been spending a lot of time working on organizations in the gene editing space. I do think once we solve some of the delivery issues and we make sure it's safe, our ability to not just improve outcome, but actually cure diseases. Obviously, we'll start with the more rare diseases, but I don't think we're far away from moving into much more common diseases. So I do think the next decade or two, in addition to all the other medicines we'll have, I do think gene editing and its cousins will play an increasing role and become one of the pillars of our standard of care to outright cure diseases that are driven by genetic dysfunction.
So let me just expand on that, because I absolutely agree, and I appreciate your enthusiasm for that as well. But when I think about us succeeding and I think about impact and unmet need, what's really interesting from my point of view is the notion of a one-time intervention with an editing strategy and modifying the disease without really any ongoing need for therapeutic treatment. One-time intervention actually could have the greatest impact in developing countries as opposed to developed countries. So if we could get a patient into a center and give this intervention, even though the public health infrastructure may not be great, think about the impact that we could have on diseases that otherwise would not be tenable if the patient had to keep coming for a lumbar puncture or a protein infusion. So as we roll this out in developed countries, I think as a community, we need to think about how to provide access in emerging markets, and the impact that we could have in 10 or 20 years when we think back on that should just be enormous.
Good point. Thank you, James.
And maybe just to elaborate further on what I said, I think I was fascinated by the discussion you had among treat early, even at the level of newborns, detecting, screening more, and cure before it happens. That's fantastic for humanity. And I would probably link that to the end of life, where this Alzheimer and neurodegenerative disease are a disaster for the whole society, because the elderly are kind of a burden, and societies will not be able, and there are in developed countries, maybe more, maybe, but there is not any familial structure and strengths to help those patients. So if we can find a way, understand what's happening in the brain and crack that barrier, and find the right animal models and the way to develop drugs for this population would be, I think, an enormous improvement for patients and their families.
Thanks. Matt, your final concluding remarks?
Yeah, I think for me, just as the last couple of decades were the years of monoclonal antibody therapy for cancer, and I think the next 20 years is going to be engineered cells as treatments for cancer in a format where it's democratizing the access to the drugs. In other words, not one CAR T cell for one patient, but maybe one form of a CAR T cell that could be given to thousands of patients through all these clever engineering techniques. And then you know, the wondrous effects of those drugs can be available on a public health scale, as opposed to a patient by patient scale.
Great. Thank you, Sabine, Matthew, David, and James. We have this very insightful discussion. Thank you again for attending, being the panelists, and we'll see you next time.
Recent advances in both embracing chemical breakthroughs and understanding biological systems have opened up new opportunities for developing drugs that target previously undruggable targets. What are the current approaches that help address the challenges of druggability? And how can we be bold in translating these approaches into major patient benefits? Join us as we showcase several innovators at the forefront of these approaches and learn about their progress in this exciting field.
I'm Laksh Adani, co-founder and CEO of Charm Therapeutics, a small molecule drug discovery company with an AI platform that is able to drug hard-to-drug targets, and we're focused in oncology. For me, druggability means the ease and potential of a protein target to be modulated, by some sort of therapeutic modality. In our case, that would be small molecules. So I think a lot of drug targets, as an industry, we've made progress against a lot of drug targets over the past, couple of decades. Most of those targets are ones where there is a druggable pocket, which might be an active site, for example, which has been designed over many cycles of evolution by nature, essentially, to bind a natural ligand. I think drugging allosteric pockets, in proteins that do not have a binding site designed by nature, which is actually most proteins, by the way, around 85% of all proteins, that's been a lot more challenging, I think, for our industry. And I think that's something where we're going to be making a lot of progress over the next decade. So our platform, I would say, is therapeutic area agnostic. Of course, there are proteins that are involved, in any therapeutic area that could potentially be, modulated by our platform. Our platform also has the potential to conduct selectivity analyses for molecules. So you could imagine taking one of your lead compounds and folding it in silico against the entire proteome to make sure that your compound wouldn't bind to any off targets. And so this would probably be useful in therapeutic areas where a high degree of selectivity is very important to avoid idiosyncratic toxicity. I think we've made very good progress on showing that the platform can deliver high quality chemical matter against, hard to drug targets. We haven't yet disclosed our targets, but hopefully we'll be in a position to do that soon. The next milestone would really be, nominating a development candidate for one of our, targets that we're working on.
Why is that we are having difficulties going after incredible targets with small molecules? Small molecules need to find their way into the protein. They need to bind. And because we don't see a pocket, we assume that the protein has no pocket, and we take modality risk. We go to something else, something that doesn't need a pocket. However, very often these proteins do have pockets. It's just that they're invisible to us. And this comes for a variety, there are two major drivers of this mismatch. And that's what our technology addresses, our technology called Resolve. The first problem that we solve is, as you know, these proteins, they're not static, they're moving, and they're assuming multiple conformations. We developed a tool that allows to group these many conformations into groups, 10, 20, 40, 3, in a totally unbiased way. We don't touch it, meaning the software will define, using the first principles of physics, how many conformations a protein assumes. Second, and that's the most important piece of our technology, and it's proprietary to us as well. If there is a pocket, if the protein has a pocket, the pocket is filled with water, just because the protein is inside the cell. But the water is not static. The water is moving, donating bonds, receiving bonds, behaving as grease. And irrespective of these energetic properties, water with these energetic properties bound to the pocket. So if I can now train virtual screenings to only look to chemical matter that shares that properties that we extracted, and we call this the hydrocophore. So when we extract the hydrocophore and train the virtual libraries to only look for that as a blueprint, guess what? I only get that. But whatever I get, which is not thousands and thousands of hits, we get dozens. It shares the properties of the water, which means it will bind. So we profoundly enriched the mechanism. And with this information in hand, we can now synthesize this in the lab, these very virtual binders that we selected, generate new hits, so generate new chemical matter, and then use the same technology to iterate again to optimize. And that's, and to tell you a sense of the productivity that this yields, we screened our first target in 2020, and we got 3 programs clinic stage in 2023. It's an incredibly efficient. So that's what the technology provides.
BridGene's platform can be utilized in any therapeutic area that requires the development of drugs for the undruggable targets. So our company focus lies in the field of oncology and the CNS, which we believe are these are therapeutic areas with the greatest unmet medical needs. So our approach offers several benefits to patients. First, we know there are more than 20,000 proteins in human proteome, with approximately 12,000 of them are being confirmed or considered to play a role in human diseases, and potentially can become a target for precision medicines. However, so far, over 90% of these proteins currently have no drugs. So BridGene has already identified small molecule ligands for over 3,000 proteins, and we aim to identify small molecule ligands for at least 10,000 proteins by the end of next year. So this can help bridge the gap between small molecule drugs and undruggable targets, provide patients with new treatments for unmet medical needs. So save life, serve patients, this is our mission, and this is something we are very excited about it. BridGene is working to unlock the full potential of our platform by attracting top talents in chemistry and in biology, And also we try to secure support from major pharmaceutical companies, investors, and also academic institutions. To maximize the technology potential, we believe the advancements in mass spectrometry and the automation of our experimental processes will play a significant role. So the company is aiming to reach several key milestones that including we will file the IND for our leading program by end of this year. This program is directly identified by AppTec protein-wide live cell screening and developed in-house. And this milestone will be the validation of our AppTec platform. Then we will advance into high-value undruggable target program to the IND enabling stage. Finally, we will establish one or two major collaborations with the large pharmaceutical companies to fully explore the potentials of our platforms.
There are two components to druggability. There's the physical structure of the target, and then there's where it actually lives in the body. And you need really to be able to have both of those things. A molecule with the ability to directly interfere with the function of the target, and that can get to the target where it lives and works. And I think both of those requirements are important. Can we open up every target in the transcriptome, every RNA, by directly targeting RNA with small molecules that then have broad biodistribution, that can go everywhere in the body, that have all those remarkable properties that we can eat them, go everywhere, seek and destroy specific targets, and can be produced cheaply en masse and made available to as many patients as possible. So that's what we're doing. What motivates us at Arrakis is to gain access to large swaths of human biology that are well understood to us today, but are simply out of reach because our drug discovery toolkit can't access those targets. How do we bring all that biology into play to create new medicines for patients? Our approach to doing that is to bring the power of medicinal chemistry to the transcriptome. Can we drug the entire transcriptome with small molecules? And of course, RNA is everywhere, and consequently, no therapeutic area is off limits. That said, we know that we can't do it all ourselves. We believe there are dozens, perhaps hundreds of medicines to be discovered off our platform, and we just can't do it all. But that's what we want. We want all of those medicines to ultimately get to patients, and that's why partnerships have been so important to us. We have two very significant collaborations today. We're working on multiple targets in their key therapeutic areas, as well as working on internal programs of interest to us. And again, we want to see as many of these things as possible get to patients. We don't really care who sells them in the end. If we can deliver all of these medicines to the world, we will have done well for everyone.
We deliver targeted protein degraders using the tools of the ADC world. And we call this approach TPD squared because it's dual targeting. One, you have the degrader target. Second, you have the antibody target for the cell type. So TPD square is quite, I think, exciting because if you think about it, think about what antibodies has done to the cytotoxic world. There are drugs in breast cancer that are ADCs that have topoisomerase 1 inhibition that was not really a breast cancer target before. It was more of a colorectal cancer target, and there's approved drugs there. But by using antibodies, you could actually use topoisomerase 1 inhibitors in breast cancer patients very effectively and safely. So we're trying to bring that sort of value addition to the degrader world using antibodies or the TPD square approach. This way, we can not only make these degraders safer, but we can actually increase the half-life. We can make it more effective. We've actually seen in cases in vitro, we've been able to increase the efficacy by 1,000-fold in terms of EC50. So TPD square approach, that approach just entered the clinic last year. We announced the first patient dose of our first TPD square asset. It's called ORM5029. It's a pertuzumab delivering a GSPT1 degrading molecular glue to breast cancer patients.
The definition of druggability in a way continues to evolve, right? As we make progress towards understanding protein function, understanding the link between a target and a disease. At STABLIX, for example, we are expanding that definition of druggability by recruiting a class of proteins called deubiquitinases, or DUBs for short, to specific targets of interest. And the outcome of that is that we are removing the chain and achieve pharmacology that way. So this concept really was not possible just a few years ago. And it really opens tremendous new possibilities for drugging a whole new class of targets. The investment we made in our platform at STABLIX, we were able to develop this exquisite knowledge in terms of the ideal DUB target pairs. And this provides us sort of an atlas map, right? This atlas can become almost programmable. So we can tell upfront, given the knowledge we have been learning from all the experiments we have done, that this target is best suited for this particular deubiquitinase or DUB. So make it programmable over time. And so that's really tremendous progress we made on that front. We have identified several new scaffolds for these DUB binders. As you know, DUBs or deubiquitinases is a notorious class known to be challenging for drug discovery because they have this class of enzymes have an active site that has a reactive cysteine. And if you react covalently with the cysteine, you will block the activity of the enzyme. And that's not really an outcome we want in our approach. So we really made tremendous progress in identifying neutral binders that allow us to recruit these DUBs towards our targets of interest. And all this together is really a significant progress in the field, given that, you know, this is a new area and is known to be very challenging.
Well, Rome was founded to make the dark genome druggable. Specifically, we're developing a proprietary data science platform using machine learning algorithms that allow us to map and quantify repetitive elements, which are sequences that make up the majority of the genome, and which have till now been understudied. The rich set of information that's obtained by our technology enables us to decipher pathways in which dysregulated repetitive elements drive disease processes and uncovers novel targets that can be addressed with all of the available modalities. We found that different classes of repetitive elements are dysregulated in particular disease conditions: cancer, autoimmune disease, neurodegenerative diseases, and aging. And we believe that targeting these elements will provide profound benefits to patients suffering with these conditions. Since these repetitive elements are typically turned off in all healthy subjects, we don't anticipate any mechanism-based adverse effects when drugging these repeat-based targets. By their very nature, repetitive elements are incredibly hard to quantify. Traditional bioinformatic tools don't do well in aligning sequences to specific places in the genome when these sequences are not unique. So most bioinformatic tools actually filter out repetitive sequences before analysis. So our data science platform is fine-tuned to annotate and quantify repetitive elements. And this technology enables us to identify which patients show dysregulated expression of these different elements, which tell us which patients we can treat with our drugs. And to realize the full potential of this precision immunology approach and precision oncology, precision neuroscience, then we need to develop biomarkers that can be used in standard clinical practice. First developmental candidate, a small molecule targeting line 1 reverse transcriptase, which is a repeat encoded enzyme. It's marching towards the clinic for autoimmune disease. Our technology enables us to identify which patients show dysregulated expression of line 1, which tells us which patients we can treat with our drugs. The next milestone are first in human studies, followed by proof of concept clinical trials in patients with lupus and SLE and CLE. There are many additional indications and other autoimmune diseases that would follow from that. So we have a number of really key milestones coming up.
Diagram Therapeutics is a biotech company focusing on the discovery and development of novel molecular glue degrader drugs. Our technology, it expands the drug target space into types of proteins that are traditionally not druggable by small molecules. Why is this? Because the molecular glue, the modality, the mechanism of action is where the requirement there is certain protein structure, certain motifs on the target protein, as opposed to activity pocket or a binder. We're looking for certain structures and motifs, and these can include transcription factors and structural proteins and phosphatase, those, and also we can also target kinases and the traditionally targetable targets. So this really opens up the accessibility of new targets. We have made a tremendous program in a little over 1 year time of our company's existence. We have, first of all, we have developed a technology platform. As I mentioned, we have a very unique compound library that has different structures from the lenalidomide and pomalidomide core. And we use that, we can discover novel targets different from the traditional targets seen in the molecular glue degraders. And we have a very extensive validation system that will help us discover new targets. So these are the platform components, and then all the AI and the machine learning. So we're building that. And with the platform, we have already generated a very robust pipeline. We have 3 programs in lead, 2 in lead optimization, 1 in preclinical development already. And we also have another, so we have a total of 20 programs. The other ones are in earlier stage, in different stage of target validation. So I think this is a tremendous progress for a new company and new technology.
For in silico methods, I would like to see methods be able to accurately find molecules that not only bind to targets that are traditionally considered hard to drug, but also modulate them functionally.
I expect to see drugs, phase 3 trials, approved medications that came from until now undruggable targets, solved by technology.
I believe that next 5 years will bring exciting advancement in the druggability space that will lead to the discovery of new, effective therapies for a variety of diseases.
Delivering complex molecules to where they need to go in the body remains an outstanding challenge. And I think that's probably where we're going to see a lot of traction over the next 5 years.
I think if people will innovate in sort of mix matching different validated technologies in a novel way to solve problems that existing modalities have difficulty solving.
I hope to see a whole generation of new medicines that take advantage of the concept of chemically induced proximity. In the next 5 years, I would hope to see compounds coming out of pipeline and in the clinic.
Technology improvements in engineering and manufacturing cell therapies would make a big difference. New delivery vehicles and gene engineering vectors would open up many doors. So a lot I'm predicting is going to happen over the next 5 years.
I think there will be more traditionally undruggable targets that will become druggable with these new modalities and new approaches.
There is growing excitement around the potential of using RNA molecules as a new class of therapeutics for diverse medical conditions. What are the critical challenges in realizing the full potential of RNA therapeutics? And how can we be bold in overcoming them? We invite you to participate in our panel of leading experts as they share their insights and discuss productive approaches to advancing RNA therapeutics.
CAMP4 is leveraging antisense oligonucleotide technology or chemistry, so ASOs commonly known, to amplify mRNA. And the way that we enable this approach is through our platform that is meant to pioneer a whole new era of transcription, that is influence of what's called regulatory RNAs. These are RNAs that arise out of the non-coding genome. For example, they arise out of enhancers and or promoters, and they act as rheostats to control protein coding genes. That is, they can influence the expression in a very specific way of protein coding genes, causing the amount of mRNA to go up or go down. Because we are studying a fundamental area of biology, that is the way genes are expressed, we essentially have a limitless amount of opportunities to be able to influence the expression of genes. So what that means is we can go after both rare and non-rare diseases. And because we are a platform company, we are doing as such. We do tend to prioritize diseases where, one, there is a strong genetic link to the disease. We look for places where we can also have clear readouts early and quickly in the clinic. And then we also think a lot about unmet need. The last thing that we think about for our technology, because we're using ASOs, is starting in tissues where delivery has been relatively de-risked. That is, where we know the chemistry can be delivered in a safe and effective way. CAMP4 has been built from the ground up. We're about 6 years old, and we've made quite a bit of progress, and we're quite proud of it. So starting from building the platform, we've now gotten to a stage where this year we intend to file, or submit, I should say, multiple INDs for diseases, for example, Dravet syndrome, which is a rare genetic epilepsy in the central nervous system, as well as for diseases that are metabolic in nature. So our first rare disease program from the liver is urea cycle disorders, which is a buildup of ammonia leading to all types of comorbidities such as neurological sequelae, vomiting, nausea, and other types of problems. So it is really a very important year for CAMP4 as we look to shift from just a discovery company into a clinical stage biotechnology company. And so that's a major milestone for CAMP4, and it's our first opportunity to prove the power of our technology to essentially be able to upregulate genes and show that they can have very meaningful therapeutic impacts.
So we've been very focused on developing treatments for chronic diseases at Okra Bio. We think the amalgamation of deep genomic profiling of human tissues to really look for fundamental pathways that drive disease in combination with more translational human tissue-based models will ultimately improve the success of finding treatments for complex chronic diseases like metabolic dysregulation or dysfunction, or diseases related to cell stress like acute on chronic or later stage liver disease. And we think that our technology platform is really well set up to do that, as particularly as the modality of siRNA begins to mature into more complex chronic diseases as well. So one of our founding theses is that human organ models or human tissue-based models are more translational than a traditional mouse model or otherwise that have been historically used in this field. So we do a lot of work on taking organs that can't be used for transplantation because they're too fatty or too old, keeping them alive on these devices outside the body and using that as a way to really study in a human relevant setting whether our medicines are having an impact on human disease. But one of the challenges, of course, in doing that is you can only keep human organs alive outside the body for a limited period of time. So you have to think about ways to measure phenotypic readouts in a 5-day window is where we've gotten to. But we think that is sufficient to really see interesting biology shine through that will ultimately enable us to do further research and give ourselves confidence in the target. We've spent the last few years really building out our platform capabilities from discovery through the validation, and we've seen huge success in that. Being able to actually keep organs consistently alive outside the body for almost a week is a huge milestone for the company, and enabling us to better test out new RNA medicines. The next big challenge we're facing is designing and executing on our clinical development plan that's coming together. So we're really excited to now transition the company from a discovery stage really through into clinical stage business.
Atheris is pioneering the development of next-generation mRNA therapeutics and vaccines for respiratory diseases and infections with high unmet needs. And our goal is really to improve patients' lives worldwide. And we move our first development candidate into the clinics in summer this year. So we at Atheris, we are accelerating the development of two main programs in our pipeline, and one with a broad therapeutic applicability for respiratory viral infections, and a rare disease with a really truly high unmet medical need. And the first mRNA candidate, that's ETH47, and this contains a messenger RNA encoding for a type 3 interferon, and this can be broadly used to treat respiratory viral infections and also exacerbations. It's active against influenza, SARS-CoV-2, RSV, and many more. And this is by activating the body's innate immune system at the site of viral entry. And the second messenger RNA candidate that we developed, ETH42, is a messenger RNA-based protein replacement therapy being developed to treat primary ciliary dyskinesia, which is a severe life-limiting genetic pulmonary disease caused by defects in motile cilia in the respiratory tract with no treatment yet. The disease is prevalent in approximately 1 out of 20,000 individuals worldwide. And we also look into developing our technologies for next generation messenger RNA vaccines with our mRNA technology, in particular the highly thermal and mechanostability that can potentially overcome the shortcomings of the currently available vaccines.
At Rana Therapeutics, we have high priority program with siRNA therapeutic modality. With siRNA approach, we can address those diseases whereby knocking down RNA or protein level, it can improve the clinical outcome. So at Rana, we have two proprietary platform technologies where we deploy our large discovery resources on. The first is definitely the delivery platform. Although hepatic delivery is our cornerstone platform, we have great resources and discovery platform in extrahepatic delivery, where everyone wants to address globally, especially on central nervous system and ophthalmology delivery platform. We have already generated great data in our human primate studies, and they will be approaching marching into clinic next year. And we want to build our proprietary delivery platform utilizing the beauty of conjugation to achieve more targeted delivery. And our second platform is an RNA modification. This part is actually as revolutionized as the first part of delivery itself. It is really those modifications that conquer the druggability problem for RNA therapeutics, from siRNA to mRNA. They all have great RNA modification that make them with druggability characteristics. And the real realm of modifications leave large space for scientists to further explore. So we at Rana, we are exploring some of the pioneering modifications to apply on siRNA molecules to increase the stability, duration, even to potentially achieve annual dosing goal as truly transformative therapeutics. And we've made great progress in all these platforms as well as the pipeline, while we will see our first liver-targeted program marching into clinic this year and next year with our extrahepatic delivery platform programs.
We work in the RNA therapeutic class known as siRNAs. Our approach is to leverage fatty acids to enable extrahepatic delivery of this particular class of molecules, siRNA. I think one of the cool things about what we've uncovered at DTX over the last 12 to 18 months is the combination of fatty acids that you select for covalent coupling to the siRNA dictates uptake and biodistribution. And so when we talk about our platform, we always stress its versatility. We're able to play in a number of novel therapeutic areas. And one of the therapeutic areas where we have a pretty differentiated approach is the peripheral nervous system. And there's an indication there called Charcot-Marie-Tooth 1A disease. It's a peripheral neuropathy driven by an extra copy of a gene called PMP22. And that gene is overexpressed in a cell type called the Schwann cell. And the Schwann cell coats your axons and is important for efficient transmission of nerve signals. There's more than 75,000 patients in the US, 150,000 if you include Europe as well, and many more if you include the entire world. And so we're very attracted to this particular indication because there's no drugs in clinical development that address the underlying genetic lesion. And in animal models, we've been able to show we can reverse the disorder. And so that's one example of an indication we can play in where we think we have the lead. The other places where we deploy the technology are neuromuscular indications. So there's a number of neuromuscular indications, DM1, DMD, FSHD. So we're generally playing in that space as well. With respect to milestones, we're about 8 to 10 months from filing our first IND application for CMT1A and are on the verge of completing some of our first GLP toxicology studies. We've made our first GMP batch of materials. So that was a pretty exciting feat for the company. And then additionally, we've generated non-human primate data across at least 4 distinct therapeutic areas with sort of different combinations of fatty acids, validating that we can knock down genes in their respective target tissues. And so, I think milestones that you can expect from us as we move into 2024 is our first dose in humans, hopefully a second IND for a second program in 2024, and then hopefully the announcement of additional programs where we can deploy our technology.
We believe we are on the cusp of a revolution in genetic medicine, and the key to unlocking that is delivery. ReCode is powering the next wave of genetic medicine through our powerful, superior, next generation lipid nanoparticle delivery platform. The precise delivery of genetic medicines to organs beyond the liver has long been the holy grail for advancing these potentially curative therapeutic strategies for a broad range of diseases. ReCode has a selective organ-targeting lipid nanoparticle platform that enables the precise delivery of a wide range of genetic cargoes to organs and cells beyond the liver. The use of these LNPs as a packaging and delivery vehicle for genetic cargoes like mRNA is well validated. But what if we could go beyond mRNA to DNA, to siRNA, and to next generation gene editing technologies? That is our goal. What's really unique about our LNPs is the ability to take a traditional 4-component lipid nanoparticle and engineer it with a differentiated 5th or 6th lipid. These sort of LNPs have a biochemically distinct lipid composition that help the body sort and direct LNPs to other targeted organs beyond the liver. In addition, we have great versatility. We can precisely and in a programmable way with both modes of administration, such as intravenous, subcutaneous, and the diversity of genetic cargoes, target these genetic cargoes to different tissues. Our lead programs are therefore in the lung with inhaled formulations of mRNA targeting primary ciliary dyskinesia and cystic fibrosis. But beyond that, the SORT LNP platform is the foundation for a pipeline of disease-modifying mRNA and gene correction-based therapeutics that are aimed at rare genetic diseases. We are expanding into areas such as muscle, CNS, and oncology indications. And the whole point of this is to transcend the limitations of today's genetic medicine delivery systems, which by and large go to the liver, so that many more patients may benefit from the disease-modifying properties of these genetic therapeutics.
So at Transline Therapeutics, we're developing therapeutics for important neurodegenerative and ocular diseases in terms of our internal pipeline. We're doing that with a unique platform technology we have for the physiological upregulation of protein expression. The platform we have, the SINAP platform, derives actually from the work our founders did on natural long non-coding RNAs some years ago, and they discovered a subset of long non-coding RNAs which bind with coding RNAs coding for proteins and serves to naturally upregulate their level of expression in the cellular environment. And we're working to take those constructs and engineer them into therapeutics. What's unique or important about these is that if you compare what we're doing to historical RNA therapeutics, they've tended to be focused on knockdown strategies, taking out proteins that you don't want to be expressed. And similarly with many monoclonal strategies. Now, there are now a class of RNA therapeutics emerging, of which we're one, focused on pushing up the level of protein expression by small amounts. In our case, we have a unique mode of action by upregulating the rate of translation rather than transcription. And that gives us, we believe, a chance to address a wide number of proteins and get small, precise levels of increase in protein expression. And that's difficult to achieve with gene therapy and many other platforms. And we think that it's important because there are many targets emerging, particularly in CNS and neurodegenerative diseases, where you need the ability to precisely upregulate a particular protein, and you need longevity of effect, precision, and safety for the long term. When I look at the way we've had to invest our time over the past couple of years, it has very much been around developing the right assay strategies to allow us to go from a very empirical mode and taking prototype candidates and really understand how we can start to improve those and give them the characteristics necessary to become therapeutics. So we've put a lot of investment in identifying and developing the right assay strategies and starting to get under the bonnet of mode of action with what we do and what our founders do. Where we are now, we're now moving into the translational phase. So we've already conducted some early preclinical experiments. And this year is going to be all about that, actually. It's all going to be about moving forwards through candidate optimization. And we're looking to be getting into some very exciting in vivo data by the back end of this year to give us preclinical proof of concepts in our lead indication, plus starting to see, we hope, some very exciting in vitro data come through for some quite important new CNS targets.
I think that there's a wave of really exciting editing technologies and mRNA and circular RNA technologies that years from now could have a major impact.
Medicine really needs to think about preventative treatment. And I think RNA is this very unique selectivity and specificity that enables it to think about a preventative treatment.
We hope to see new small biotech companies with really transformative ideas around using messenger RNA and new technology that start to come of age, and new product candidates that can benefit the broad patient population worldwide.
I would expect to see the advancement of RNA chemistry to further improve the druggability of the therapeutics, to bring not just siRNA, but also other RNA modalities into clinic or into approval.
Over the next 5 years, we would hope to see the promising preclinical data from DTX and other companies with innovative RNA delivery technologies translating into clinical proof of concept and providing some hope to patients who previously had no treatment options.
I'm particularly excited about the collaboration in the next 5 years as we bring together delivery and cargoes to accelerate toward the clinic with these new therapeutics for patients.
I think We're going to see a lot more new submodalities progress into clinic and to get to clinical POC. So that will be building on that pioneering work done in the sRNA and ASO fields.
Cell and gene therapies have significant potential to provide more targeted and personalized treatment options that can lead to improved outcomes for patients. What therapeutic areas are likely to see the biggest impact of these therapies? And what is the current progress in this field? Join our panel of innovators to learn about their approaches to advancing these therapies and how they could boldly transform the way we treat a range of diseases.
Yeah, Sonoma Bio was based on work that my co-founders and I started in an academic setting, looking at the possibility of using a novel cell population called regulatory T cells, or Tregs, to try to treat diseases such as autoimmune diseases, organ transplant rejection, and other inflammatory diseases. The modality is really based on cell therapy as it's been pioneered in the cancer space, but using a very different cell population. These Tregs are designed to shut down or induce a tolerogenic environment so that patients are not continuing to have this ongoing autoinflammatory disease. It is certainly cutting edge. As many of you know, cell therapy so far has been largely limited to the treatment of cancers, and the idea of being able to use a novel cell population to treat autoimmune diseases is something that has been tested preclinically and to some extent in early academic clinical studies. So as a company, we think that our differentiating activities really are related to the platform itself and the cells that we're using. After decades of experience in working with these cells, we feel we have the best ability to identify, isolate pure populations, to expand them selectively and give them specificity to tackle a specific autoimmune or autoinflammatory disease. We're hoping that this will be a one and done treatment where the cells, being a living therapy, will be able to get to the site of inflammation, proliferate, get activated, and treat the disease over a long-lasting period of time. And key milestone for us is to get these cells into patients in this disease and demonstrate proof of concept that this therapy can lead to moderation of disease progression and can do that in a safe and durable way. So we've really made significant progress so far. We've entered the clinic with 5301, our T effector conditioning biologic, and we've been breaking new ground on our state-of-the-art R&D and manufacturing center, both big milestones for the company. We also have presented early data highlighting the potential for our Treg therapy in RA, and expect that this lead program will be in the clinic this year.
Intergalactic was formed to overcome the limitations of viral-based gene therapy and develop best-in-class non-viral alternative. We are advancing a revolutionary approach that bypasses historical drawbacks to gene therapy, while broadly unlocking the potential of non-viral gene therapies for a wide range of diseases. Specifically, our proprietary integrated platform comprises of three core pillars to bring local non-viral gene therapies into reality and transform treatment paradigms. So the first pillar, our scientists have developed a versatile and programmable non-viral gene therapy technology called C3DNA, which stands for covalently close and circular DNA. One of the key benefits of C3DNA is that it allows for the design and delivery of DNA vectors beyond the size limits of current viral delivery system. Also, C3DNA is precisely engineered using synthetic biology tools to assemble modular elements into a closed loop, allowing it to be taken up by cells and expressed without insertion into the host genome. And because ctDNA does not use unintended viral and bacterial sequences, it avoids triggering an immune response and allows for redosing. They are just some of the potentially groundbreaking benefits of our proprietary technology. Second pillar of our platform is the precise tunable system for local delivery of ctDNA called COMET. It stands for cellular delivery of genetic material by electrotransfer. So enabling non-viral DNA applications, and it engineers to work with different types of ctDNA therapeutic payloads and target specific tissue. Last but not least, the third pillar is an innovative, scalable manufacturing process that really leverages the standard and widely available manufacturing infrastructures and enables optimal CMC scalability and economics. All combined, this fully integrated platform has significant potential benefits for patient safety and efficacy. It really offers the versatility to treat a broad range of historically untreatable diseases with simple, fast, and cost-efficient production to make gene therapy more accessible. This transformational approach really shows that Intergalactic has cracked the code to advance a potentially safer, more effective, more versatile, and more accessible solution to expand gene therapy universe and bring life-changing treatments to patients in need.
Anoko works with TCR-T, which builds on the successes of CAR-Ts, which have had major impact in a select number of hematological malignancies, but so far no success in solid tumors, which represent about 90% of all cancers. A key reason for this lack of success is a lack of genuinely tumor-specific cancer targets at the surface of cells that make up solid tumors, which you need for the current CAR targeting modality. For T-cell therapies, the TCR targeting modality that Anoko works with solves for the lack of CAR T-cell surface targets. Now, TCRs themselves are a natural targeting system for T-cells and detect disease targets derived from within cancer cells through presentation of targets in a complex called an MHC. There's a key challenge in accessing MHC-presented targets, and that across the human population, there are thousands of MHC variants that present targets differently, meaning that even for the same target, say a specific KRAS mutation, different patient groups will present the mutant target differently, and thus you need the different targeting TCR for their cell therapy. Ultimately, this means we're looking at a therapeutic modality that will comprise product franchises that span both targets and MHCs in a highly personalized manner. Now, Anoko's platform is based around a concept of precision biology, and we've built a range of unique precision cellular assays to unlock that rich cancer-specific target space, deploying libraries of TCRs that can be customized to specific patient segments that share target and MHC expression. Basically, we're aiming to unlock the transformational promise of TCR-based cell therapies and delivering products for treatment of solid tumors for a number of targets across a large number of patient segments. We've used the platform to generate dozens of TCR assets today, and the next major milestone is moving a selection of those assets into clinic, which is on the agenda for 2024. We'll be doing this with our own autologous manufacturing approach within our own GMP infrastructure, and so there are a number of regulatory milestones to move through to that first in human target next year.
Yeah, we're a startup biotechnology company in the preclinical stage. We're a spin out of the University of Pennsylvania. Our scientific founder is Dr. Sar Gill, who is a professor at Penn. Sar is an immuno-oncologist at the University of Pennsylvania, and his technology that we licensed allows us to begin to think about making CAR T cells actually in vivo as opposed to the standard ex vivo that's in the field today. So the whole concept from Sar's perspective and ours is that we want patients to be able to access this technology quickly and easily as they become patients in his clinic, which is mainly a clinic that treats hematologic cancers. So the technology that we have invented here at Interius allows us to target cells inside the body. So we can take any particular cell, and let's take a T cell as the example. We can inject our vector technology and it goes directly to that T cell, reprograms that T cell and turns it into whatever we really want it to be. The idea behind being able to create CAR cells in vivo is that we can take this vector, inject it directly into the human body, and have that vector turn that T cell into a CAR cell. So in essence, we're avoiding all of the ex vivo manipulations that normally occur in the manufacture of CAR cells. For example, the patient would normally go to the clinic, have a process called apheresis, where all of their circulating cells are packaged up into a bag and moved over to a laboratory where then they are transduced, turned into CARs, and then re-infuse that cell population back into the patient. Meanwhile, the patient has to be immunosuppressed with chemotherapy. In our technology, we completely eliminate all those steps that I just went through and are simply doing it directly in the human body without any chemotherapy whatsoever. So this is a real win for patients because they don't have to have the chemotherapy, and this can literally be done as an outpatient as opposed to having to be in the hospital. So we think it's a really innovative way to approach this new therapy for patients with hematologic malignancies.
At Triumvira, we are developing autologous and allogeneic approaches based on our technology platform known as TAC, or T-cell antigen coupler, which is a unique way of activating T cells when they need to be activated, but also silencing T cells when they don't need to be activated, which is right now the biggest challenge with CAR T therapies. CAR Ts are either on or very on. There's no off switch to a CAR. That's how they're built. So they start producing cytokines even when you manufacture them already. So these poor CAR T cells have to run a marathon, especially that's very difficult in the solid tumor setting where you need to have very strong and fit cells to make it to the solid tumor, which is different than with lymphoma and leukemias. Our TAC technology has given our TAC T cells some ideal characteristics for adoptive cell therapy. So therefore we truly think that we can address the major challenges in this field of lack of exhaustion, long persistence, good penetration to solid tumors. We have shown now in the clinic that it's very safe, and also we see some great initial signs of efficacy, which is very encouraging. Our HER2 phase 1/2 program is ending the dose escalation, the phase 1 part of the study. We hope to have a recommended phase 2 dose soon. Then we'll go into expansion cohorts. And not only are we looking at different tumor types, but we're also looking at, can we redose, which is another issue right now in cell therapy in solid tumors. Secondly, can we go even higher with our dose, go to the next dose level? So that is something that we are discussing with the oncologists, the principal investigators in the study. And thirdly, we will be starting a second half this year, a combination of our TAC T cells directed against HER2 with pembrolizumab, which is obviously a PD-1 inhibitor, which we truly think there's a good scientific rationale why combining cell therapy with PD-1 inhibitors, makes sense. We're also excited to see what that does in patients.
Ivorna was founded in 2018 with the focus of bringing cell therapy to the field of autoimmune disease. We're focusing on deep B-cell depletion because we feel that there are many patients with autoimmune disease that would benefit from deep tissue-based B-cell depletion. So over the last years, we have perfected that method and really are now going into the clinic. We are a clinical stage company with our KY101 bringing cell therapy to autoimmune disease patients. I think what's very exciting to us, cell therapy is now moving from oncology, from a niche indication in blood cancers over to autoimmune disease applications where many more patients will benefit from getting access to this change in medicine. So moving into autoimmune disease, obviously a different safety profile will be required, and definitely a different patient experience will be required. So an oncology patient on the one hand and an autoimmune patient on the other hand are very, very different patient segments where we need to work through these elements in cell therapy. We're very excited about working with the NIH. We in-licensed our construct from the NIH, and recently have had FDA clearance of our lead compound, Kyverna 101. But we're also having a lot of focus on bringing an allogeneic solution to the clinic. Together with our partner, we're developing an allogeneic approach with our Kyverna 201. So Kyverna is firing in all full cylinders, bringing cell therapy to patients with autoimmune disease.
We are a gene editing company, a next generation editing company co-founded by Fong Zhang and David Wall. As a company, we focus on in vivo approaches for treating rare and other genetic diseases. And we are focused in the CNS and in the liver diseases. And that is our core focus. We partner in other areas. We have partnered in the ex vivo cell therapy space to enable companies to have access to our therapy. So gene editing, I think, we're in the very early days of editing as an industry. This new modality, I think, is going to really revolutionize the way we think about treating genetic disease, because we've now changed the paradigm to being able to treat genetic disease at the root cause of the disease. So if we can actually alter and change the underlying DNA pathology to fix it, we can provide that functional cure to a patient. And I think that's going to revolutionize the way we think about treating genetic diseases today and for the coming decades. I think this technology is going to be around for a long, long time, and will really fundamentally change the way we think about genetic disease and the way we treat and really impact patients' lives. So for Arbor Bio, our goal is to enter the clinic as soon as possible. Our lead program is in primary hyperoxaluria, which is a genetic disease that primarily impacts genetic basis is in the liver, where there's an inability to metabolize a particular substrate. That substrate then goes to the kidney and causes massive kidney stones and destroys the kidneys in patients when they're young adults. So it's a very devastating disease. So being able to prevent the buildup of that toxic substrate is what our therapy is designed to do. Our goal is to enter the clinic as soon as possible, with a goal to file our initial CTA, or clinical trial application, sometime in the next 9 to 12 months. I think that the goal would be to, enter the clinic, hopefully treat these patients and deliver a one-time treatment that can provide significant benefits.
Tessa Therapeutics is a clinical stage biotechnology company. We're developing a portfolio of next generation cell therapies for cancer. Our off-the-shelf or allogeneic cell therapy has several advantages in comparison with conventional autologous CAR-T technology. As you know, autologous CAR-T technology, it's already in the market. It takes a long time because we have to use individual patients' T cells and modify them. So we believe off the shelf could be truly transformational and could be the next frontier in cancer treatment. So Tessa is developing a unique and potentially transformational allogeneic off-the-shelf CD30 CAR-modified Epstein-Barr virus-specific T cell platform. That's a very long description. But this platform is capable of targeting a broad range of malignancies, and that's our near-term clinical focus. Our lead clinical program, which we call TP11X, which is this off-the-shelf CD30 CAR modified EBV-ST, is currently being developed for the treatment of Hodgkin's lymphoma. As you may know, Hodgkin's lymphoma is highly expressing CD30, so this is why we start there. So we're doing a phase 1 clinical trial right now with TP11X, That's still ongoing, but we have shared interim data at the last ASH conference in New Orleans, showing 79% overall response rate with 43% complete response, and that was achieved in 14 heavily pretreated patients. So they were all relapsed refractory. So this is a very good result in terms of efficacy. What's also remarkable is that it was very well tolerated. So we did not have any dose-limiting toxicities, including no evidence of graft versus host disease or neurotoxicity or any higher than grade 3 cytokine release syndrome. Based on this data, we believe that TT11X may have potential in not just Hodgkin's lymphoma, but also in non-Hodgkin's lymphoma. And we also have an intent to develop this TT11X EBVST platform for additional cancer indications, including solid tumors. I think a key differentiator for us is that at the efficacy data that of course still needs to be seen at the end of the trial, the safety data seems to be very, very good. And that actually enables us to even increase the dose further and then further improve the durability.
At Regeneron, we are dedicated to developing the next generation gene therapies to treat ophthalmic diseases with high unmet medical needs, while also leveraging our innovative technology platforms to partner with the leading biopharma players and to develop AAV-based gene therapies for retina, ophthalmology, neuroscience, liver, cardiac, and etc. Regeneron has 3 next generation technology platforms, and that is really designed to overcome the limitations of the existing AAV-based gene therapies. The first platform is a VG-AV. It's a novel AAV capsids platform, and we designed this platform in order to really enable this capsid to have a superior transduction efficiency and have the capability also to potentially cross biological barriers. The second technology platform is called REVERT, and this is reconstitution via the mRNA transplicing. So REVERT is designed really to overcome the limitations of the AAV delivery, which is probably about 4.7 kb. And the differentiation, or is actually the breakthrough tool that we developed at REVERT, is to go through this, to reconstitute at mRNA level by the dual AAV vector approach. The third technology platform is a gene transactivation. And this is actually designed also to really deliver the CRISPR-Cas-based AAV gene therapies to modulate one or multiple of the genes simultaneously. At Regeneron's 2 in-house pipeline programs are in development for the treatment of inherited retinal diseases. So our first program is used also the VG vectors for the retinitis pigmentosa. And the second program that we are developing is using our both technology platforms. Is VG-AAV capsid and also our REVERT reconstitution via the mRNA transplicing. So we're using these 2 technologies to develop a program for Stargardt diseases. Our advantage using our innovation technologies is that we can deliver also provide these treatment options efficiently and safely, potentially to the patients, also via the intravitreal injections. And we believe that is also could be a good way to really also have good safety profiles while we are also achieving the efficacy for the patients.
I think the biologic revolution of the last couple of decades will make the 21st century really similarly advancing medicine in the way we changed industrial processes in the last century. And so because of that, I think cell and gene therapy are the fields that are going to lead the way.
We'll see the promise of non-viral gene therapy, my hope is that it opens the doors to more and more therapies, getting them approved for patients.
Therapies that recruit or retarget T-cell immunity are going to achieve a giant leap forward in the coming years. There's one thing I'd really like to see more of, and that is deep collaborations between peer biotechs, basically collaborating to make class-leading products.
The gene therapy area, every single day, there's a new paper, a new breakthrough that's coming along, that the future is just extremely bright for that technology.
I hope that both autologous as well as allogeneic cell source and manufacturing are well established in 5 years from now.
We are in front of a very exciting phase of cell therapy, where cell therapy moves from oncology setting into an autoimmune disease setting. So a very exciting moment in medicine as cell therapy goes broad.
The next 5 years would be fantastic to continue to see novel cell therapies approved and or demonstrating significant clinical benefits in patients, not only in the hematological malignancies, but also in solid tumors.
I think there will be a massive shift over the next decades in terms of using cell therapy, not just in late stage, but also in earlier stage.
We hope actually in the next 5 years, we can see really our potential to really validate the progress of next generation technologies in the clinical to deliver really the efficient, effective, and safe products to the patients.
Please welcome Richard Saul, Head of Boston Office and Senior Advisor of Strategic Initiatives, WuXi AppTec, who will close out our event.
As we come to the end of our second episode of the WuXi AppTec Bold series on innovation that matters, we want to express our deepest gratitude to all of you for joining us in this journey of inspiration and exploration. We're deeply appreciative to our esteemed speakers who have been pushing the boundaries of drug discovery and development with their bold thinking and actions. Their keen insights have been a guiding light for all of us as we navigate a year filled with challenges and opportunities. We also want to thank our audience who have joined us from all parts of the globe. Your continued support in our mission to advance breakthroughs for patients is deeply appreciated. At WuXi AppTec, we believe that being bold is not just a mindset, it also is about fostering collaboration and a call to action. We invite all of our stakeholders in the healthcare ecosystem to join us in this pursuit of bold innovation. Let's dare to imagine and create solutions that can make a real difference to the lives of patients worldwide, leading to a future where every drug can be made and every disease can be treated. As we conclude this event, we thank you for being part of this journey with us and look forward to seeing all of you at the next episode of the WuXi AppTec Bold series. Thank you.